Welcome to the 8th Annual Seattle Area.
The Seattle Epidemiologic Research and Information Center, in collaboration with the Department of Veterans Affairs, the VA Employee Education System, and the University of Washington Departments of Epidemiology and Biostatistics, presents the 8th Annual Epidemiology, Biostatistics, and Clinical Research Methods Summer Session.
Welcome to the 8th Annual Seattle ERIC Epidemiology, Biostatistics, and Clinical Research Methods Summer Session. The title of today's course is Genetic Epidemiology. The speaker is Dr. Karen Edwards. She is an associate professor in the Department of Epidemiology and the Institute for Public Health Genetics, and the director of the Center for Genomics and Public Health at the University of Washington. And this lecture will be on commingling analysis. Dr. Edwards.
Thank you. So as you all know, commingling analysis is a method to help you determine whether or not the trait that you're interested in may be under genetic influences. And as we said, when we're thinking about genetic influences, there are really two different possibilities that we need to consider. The first is that the gene is a single major gene that has a major effect on the quantitative trait. The other possibility is that it's polygenic. And with polygenic inheritance, what we're talking about is many genes, each with small additive effects that contribute to the variation in a quantitative trait. So if we think about commingling analysis and we think about single major gene effects, what we're looking at, I'm going to switch over here to this. Oh, that was good. That was a smooth transition. If we have a single major gene, what we might expect in terms of the distribution of our quantitative trait, distribution of the quantitative trait here, that if it's a single major gene, we could imagine that each of the genotypic classes, in this case we have 3 different genotypic classes, could contribute to the variation in the trait. And we might expect that each of these genotypic classes corresponded to one of these underlying normal distributions. So the idea behind commingling analysis is that we try to model the effects and pick up these commingled subdistributions. That's where the term commingling analysis comes from. Now the other type of effect that we might see when we're looking at a commingling analysis, let's just take a trait that actually is normally distributed. Now, I don't know of very many biological traits that have a normal distribution, but we can pretend there's something out there. I don't know. Certainly wouldn't be cholesterol levels. Has anybody ever seen a trait that's normally distributed? Biologic trait? Okay, well, we'll pretend there is one. So this would be some trait, let's call it X, and this would be the frequency here. If a trait is truly normally distributed, this could indicate that what we're looking at is a polygenic mode of inheritance. And we'll talk a little bit more about this when we talk about twin studies and other things. But the idea here is that if you have many genes, each with small additive effects, you still have the individual genotypic classes. But I'll do this kind of quickly and we'll talk about it more next time. Is you could imagine that there are many, many, many genotypic classes which contribute additively to the variation in this phenotype. The more genes that actually contribute to the variation, the more normally distributed this is going to be. Okay? So if something is truly polygenic, it is very likely to have a normal distribution like this. Now the problem is frequently that we have a distribution that has some skewing in it. You know, maybe not a lot of skewing, but it has some. When you're doing traditional epidemiologic work and you have a variable that has a skewed distribution, and we're talking about skewing to the right, what do you generally do to that variable before you do any other analyses? You probably log transform it, right? Purpose of log transformation is to pull that tail in and make this look more normally distributed. So let's just say as epidemiologists, that's what we do. We see a skewed trait, we log transform it, and then we think about using that for our genetic analyses. Do you think that's a good idea? Now, this is one of those things that it's kind of a mixed bag. If this is truly influenced by a single major gene, and we could imagine that this little bump out here is that one genotypic class, let's say our AA homozygotes. If we log transform this and pull that tail in and make this more normally distributed, essentially what you're doing is you're squishing together all those underlying subdistributions. What you're trying to do in commingling analysis is to distinguish each of those underlying normal distributions. If you put them all, squeeze them together so that there's more overlap, it is going to be really difficult to actually discriminate any of those underlying subdistributions. So in fact, with commingling analysis, if you log transform your trait, and this actually relates to a lot of the other genetic analyses you're going to do, you may actually be taking this genotypic class that's of interest to you, putting it together and making it more difficult to see distinct subclasses. Okay? So this is one of the first dilemmas we get to in genetic epidemiology that we usually don't have to worry about. We just log transform these things in traditional epidemiologic studies, and we need to think twice about doing that in this type of an approach. The ideal situation for a commingling analysis is when you have a distribution has a big old huge tail in it. And you can imagine, now thinking as genetic epidemiologists, that you've got three underlying subdistributions here. The more spread you have between the underlying subdistributions, the easier it is to detect each of these individual subdistributions. Okay? So as things come closer together and there's more overlap between the subdistributions, it's difficult to detect that. So one of the biggest issues for commingling analysis is power. And the factor that influences power the most is how skewed this distribution is. And that relates to the fact that the better separation you have between these underlying distributions, the easier it is to detect those. Okay? So as I said, our first dilemma is whether or not to log transform. If we do, you may be reducing the power to distinguish between these underlying subdistributions. Okay? So what do most people do? They do it both ways. They see what happens. It may be that in the untransformed trait you've got three distributions, and when you transform it may be that you only see two subdistributions. Okay? This is really not the best way to determine what the mode of inheritance is, so maybe it doesn't matter to you. But really what we're trying to do with commingling analysis is to say, can we detect more than one subdistribution? That's really what we're interested in here. If it's polygenic, one distribution, that's not good. If it's two or more subdistributions, we would take that as evidence consistent with a single major gene. But that log transformation, if we're pulling something in and making it look more normally distributed, we might mistakenly take that as a polygenic type of trait, okay? Which is not ideal for most of the other analyses that we're going to follow up with. So again, you can see even with something as simple as commingling analysis, there are a lot of decisions that we need to make, and we need to think very carefully about the phenotype. Remember, that's the lesson for today, tomorrow, and Wednesday, is to think very carefully about the phenotype. Now, here we've only talked about log transforming the trait, but you can imagine, too, adjusting for other things like environmental factors. Okay? And again, you have to think very carefully about what you're doing when you adjust something, especially when you're looking for genetic influences. You have to be careful that what you're adjusting for is not part of the phenotype. For example, if you have traits that are correlated with each other, it is possible that the reason why the traits are correlated is because there is a single major gene that underlies the variation in both traits. If you adjust for the correlated trait, what you may actually be doing is adjusting away any evidence for your genetic influences. So in genetic epi, we've got to think very carefully about what we adjust for, how we transform the data, and what our hypotheses are in terms of the underlying genetic influences. So let me just introduce one more term to you, and that is pleiotropy. So pleiotropy refers to the fact when we have, in the simplest case, one single gene. It can be many genes, it can also be polygenic, but just think of the case where we have one single gene that influences the variation in two traits simultaneously. So the way that we can think about that is, let's say we have a gene here, And body mass index, and let's think of something else what's correlated with BMI. What trait is, well, just about everything is correlated with BMI. Okay, what was that?
Blood pressure.
Blood pressure, that's a good one. Okay, so we know that BMI and blood pressure have some degree of correlation between them. What is the correlation between blood pressure and BMI, like 0.6, 0.7? It's pretty strong. Okay, but then that's probably true of many of our biologic traits. But BMI and blood pressure we know are correlated with each other. The question is, why are they correlated? Are they correlated because they share common genetic influences, or are they correlated because of other environmental factors? Well, the answer is probably both. But if you're doing a genetic association study, and let's say you're interested in identifying this gene that influences body mass index, and you adjust for blood pressure, what are you doing? You could actually be adjusting out part of the variation that's due to this gene, and you may actually not be able to detect this gene because you've adjusted part of the variation. Now, what makes genetic epi interesting is it's probably likely, we just won't draw the environmental factors, although we know they're there, that there are at least a couple of genes that influence body mass index. So this is why thinking about your phenotype, thinking about what it is you're trying to do becomes very important. If you are interested in identifying the gene that influences the covariation in body mass index and blood pressure, this is really the gene you want. Okay? So one way that you might be able to detect that particular gene is to think about some sort of joint definition that takes into account both of these traits. Okay? Some sort of bivariate analysis. Whereas if you were in the situation and you adjusted for blood pressure, you're probably identifying this gene, actually. Okay? Now, I put this up here now because this all relates to thinking about our phenotype and thinking about working in complex diseases. That oftentimes we're not really distinguishing between these different types of underlying genetic mechanisms. And if you sit down and think about it, you might be able to form your hypotheses more specifically so that you have a better chance of detecting one or these other genes, depending on what your focus is on. If you go in with something that's very heterogeneous, just like in traditional epidemiologic studies, you're not going to be able to see the effects. And this is particularly true when we're talking about genes that tend to have smaller effects anyway. That you've got to get your phenotype defined as cleanly as possible so that it's as homogeneous as possible in terms of the underlying genetic effects influencing it. Okay, let's take an example to make this a little bit more clear. You can see it's about as clear as dishwater right now. So let's say that you've got diabetes. Let's think back 20, 30 years ago, maybe it was longer than that, before we really discriminated between type 1 and type 2 diabetes. So let's say you just took all cases of diabetes and you said you were going to find the gene for diabetes. Well, first of all, it's too heterogeneous. You're mixing in at least 2 different forms of diabetes in one pot. And if you think about it, the genetic influences are going to be very different between those 2 forms of diabetes. So if you put it all together, it's going to be very difficult to tease out any genetic effect because it's too heterogeneous. This is going to be true of heart disease, essentially all of the common chronic conditions, asthma, obesity, you name them, and it's going to be the same situation. What we have found, though, is that by thinking about your phenotype and trying to define more homogeneous subclasses, you're going to increase your chances of detecting genes. And let's take the diabetes example. Some of the first genes that were identified for diabetes relate to MODY, which is maturity onset diabetes of the young. Frequently, with a condition that we're looking at, the early onset forms are the ones that have the strongest genetic influences. And so if you look at MODY, that's essentially a type 2 diabetes with a very early age of onset. We tend to think of MODY as a relatively homogeneous subset of type 2 diabetes. But in fact, I think at last count, 6 or 7 different genes have been identified for MODY. So something that we think of as a very small part of type 2 diabetes is also very genetically heterogeneous. So you can imagine then what the more common adult form of type 2 diabetes is like. If we've identified 6 or so genes for MODY, how many are there that contribute to, the remainder of type 2 diabetes? So what our challenge is in thinking about all these complex conditions is how can we take that big heterogeneous group that's probably many different forms or many different reasons for having type 2 diabetes and identify different subtypes that we can use for our genetic association studies. Now, if we knew that, obviously our job would be easy, but this is where you need to think creatively and think about the condition that you're familiar with. Are there clinical characteristics that discriminate one form from a slightly different form, and focus in on those? If it's a condition that nobody's looked at before, your best bet is to think about age of onset and select cases that occur at an earlier age. And we can give a number of examples where that has been successful. Diabetes, Alzheimer's disease, breast cancer. BRCA1 was first identified by looking at families and observing that a subset of the families had very early age of onset. And it was in those early age of onset families where BRCA1 was first identified. So think about your condition, think about age of onset. If there's a way that you can select earlier cases, those would probably be your best bet for your first shot at genetic studies. But then remember that that only gets you probably a small subset of the overall disease. Then the task is to take the remaining cases, narrow it down until you identify the different genetic factors that contribute to the disease. So this all relates to thinking about our phenotype, maybe using a commingling analysis or something else like that to try and identify more homogeneous subgroups. And when I say homogeneous here, I'm thinking homogeneous in terms of etiology and in particular genetic effects. So that really is our goal and the challenge in genetic epidemiology when we're thinking about the phenotype. Don't throw everything in there together. You're never going to find it. In genetic epidemiology, bigger is not always better, meaning sample size. As epidemiologists, we hate throwing out data. We hate to take our big data set and just look at a small subset. But when you think about it from a genetic standpoint, if that smaller subset is caused by a single gene, you're going to have a much better chance of detecting that gene using the cleaner phenotype and cleaner set of families or cases, whichever the case may be. Okay? So that's my soapbox, but that really is one of the major take-home messages you guys need to think about when you're doing your studies. And it is very difficult to do, but I'll say it again and again throughout this week so that by the time you leave here, that's what you're going to be thinking about. Okay, so that was a long-winded way to again reinforce that our phenotype is important. So I think we're going to go back to the slides now. Okay, so we've talked about this a little bit, and we'll just go over it one more time. As we said, when we're thinking about quantitative traits, and the example we're using here now is body weight in kilograms instead of BMI, most traits have a distribution that looks somewhat normal, but generally there's some skewing in the distribution. If the trait truly is a normal distribution, you should think two possibilities. One is that it's polygenic, which is not a good thing, or two, that the underlying subdistributions are very close together. And if that's the case, you're going to have difficulty in separating out those underlying subdistributions. Okay? And that may result, if you've log transformed the trait and brought the tail in, you may be making it more difficult to actually see the underlying commingled subdistributions. Okay, so as we said, this is what many of our biologic traits look like, is there is some skewing in the distribution. Most of our biologic traits have some skewing in the tail to the right, generally the higher ends of the distribution. And it's probably not in your best interest to immediately log transform these traits before you run any genetic analyses on them. Okay, and then we talked about if we're thinking single major genes and we're talking about one gene with two alleles, that we can have a couple of possibilities in terms of the underlying subdistributions. If we see a bimodal distribution, this could be consistent with either dominant or recessive effects, okay? Because either dominant or recessive will result in two subdistributions. Whereas if we actually have evidence for 3 subdistributions, it could be that it's inherited in a codominant fashion, where each of our genotypic classes corresponds to one of these underlying subdistributions. Okay. The other thing that is a little useful here, but not something that you're really going to put a lot of weight on, is that the height and essentially the area under the curve of these subdistributions relate to how frequent the different alleles are. Okay. But you're not really going to use this to quantify allele frequencies at this point. But just to keep in mind, it'll give you an idea of the relative frequency of the different alleles. If we go back to this bimodal distribution, and let's just pretend in this case that the trait that we're looking at had a recessive effect, you would imagine that in this subdistribution, which would be, for example, using our example that we used earlier, the AA homozygotes, little a, in this distribution, and then this distribution correspond to the heterozygotes and the big A homozygotes, you could tell relatively how frequent those alleles are, given the size and proportion of individuals in each of these subdistributions. But again, this will just give you a relative idea. If this subdistribution, the little one over here, was very, very, very small, you would know that that allele frequency was very rare for that particular small a allele. Okay? And it would just give you an idea of what your sample size might need to be to detect this in your next set of studies. If that's very rare, you're going to need a much larger sample size to make sure that you've got enough of those individuals. Okay? So we don't actually estimate allele frequencies using this approach, but it gives you a rough idea of what those allele frequencies might be. All right. So now let's talk a little bit about how we're actually going to do the commingling analysis. And we'll start out with our hypotheses first. So as you've probably gathered, what we're doing is we're trying to discriminate whether our distribution in our trait fits a bimodal distribution, a trimodal distribution, or is it just a single normal distribution? So what we're trying to do is determine whether or not a mixture of 2 or more normal distributions fit the data better than 1. Okay, so in this case where we're looking for 2 distributions, is our null hypothesis is that the mean of the one distribution, so imagine that picture where we have 2 subdistributions, and we're saying, is the mean of distribution 1 equal to the mean of distribution 2? If those 2 things are equal, it's the same distribution. If the mean of distribution 1 is different from the mean of distribution 2, then we're saying we have 2 separate subdistributions. Okay. Just a simple way to think about it. If the means are the same, it's one distribution. If they're different, it's 2 or more subdistributions. And as I said, the farther apart or the more space we have between those subdistributions, the easier it is to detect significant effects. Okay? So if we reject the null hypothesis here, and we say that we have 2 different subdistributions, what would our interpretation of that result be? Okay. We could say that there is evidence for a single major gene that's consistent with either dominant or recessive effects. Now, I try to use the word consistent, suggestive, because remember, this is not definitive proof of an underlying gene. We've got no genotypic data here. It could just as easily be environmental effects, but from our perspective, we're saying it's consistent with, but it is not proof of a single major gene. Okay? And that will also be another common message for the next couple of days. Consistent with, but not definitive proof of. We don't get to that point until about Friday in this class. Okay? And even then, it's never definitive in epidemiology. Okay, so if we have evidence that there are 2 distributions, then we go on and fit a 3rd distribution and see whether or not that is significantly better than 2 distributions. Okay, so we're working our way up. And then the same thing here. In this case, we're saying, is there 2 distributions or are there 3? If there's evidence for 3 distributions, we would say, well, now that's consistent with a single major gene that's inherited in a codominant fashion. Okay, not definitive proof of, but consistent with. Okay. So we already did the interpretation here. Keep in mind with most of these preliminary things that we're going to be, I shouldn't say preliminary, with most of these approaches we're going to be talking about for the next 3 days, these are nice pieces of preliminary evidence that you have genetic influences. But with all of these things, you're going to want to follow up with more powerful methods and more definitive methods where you can rule out some of these other possibilities like environmental effects, like polygenic inheritance, etc. Okay? So let's say that we go through and do our commingling analysis, and let's say that we've got 10 different traits, 2 of them look very promising, and we decide to follow up with those particular phenotypes. Some of the things that we might do next, which will lead us into our next couple of days, would be things like segregation analysis, which you will find is very similar to commingling analysis, but now using family data. So it's stronger evidence for genetic influences. We may also want to use our twin data or some other types of familial aggregation studies. But you notice that we're moving from unrelated individuals now into using data from related individuals. Okay? So these commingling analysis done in unrelated individuals, to get more definitive or stronger proof, we're going to need to move into studies where we actually have at least 2 related people, if not more. Okay? So most genetic studies, other than our case control association studies, require that you have at least 2 related people. Could be something like a SIB pair or any other type of relative pair. Okay? This is why commingling analysis is inexpensive, because it doesn't require us to have data on related individuals. All right. So you'll find when we start talking about all of these different epi methods that when you first look at these things, they look quite foreign to you, and it looks like we're just estimating tons and tons of parameters, and it's very confusing, especially when we get to segregation analyses. But what you'll find is that there are actually, in most of these approaches, a limited number of parameters that we manipulate to make different models. Okay? Commingling analysis is pretty straightforward, and again, one of the reasons we start with this is it helps us think about the phenotype. But then when we move on to things like segregation analyses, we're estimating many of the same parameters, and things should start looking familiar to you as we go through the week. Okay. So our assumptions then with the commingling analysis is that even though the overall distribution of the trait may be skewed, each of the underlying subdistributions is normal. Okay. The parameters that we estimate in commingling analysis are the means, so the mean of each of those subdistributions, the standard deviation, and we'll talk about this one in a second, and then the proportion of individuals underlying each of those subdistributions. So these are really the only parameters we're estimating in this model. Okay? So it's not too complicated. In terms of the common standard deviation, what we're saying is that each of those underlying subdistributions has the same standard deviation. We're not estimating different standard deviations for each of the subdistributions. You can, but it requires you to have a lot more data to do that. Okay. So these are the parameters that we're going to be looking at. Now, for many of the genetic epi approaches that we're going to use, we use maximum likelihood methods to estimate the parameters and also to get the likelihood. One of the things you'll find in genetic epidemiology, and this may be true of other things, but I'm just not as familiar with other areas, is that when you're using maximum likelihood approaches, one of the most important things is to make sure that your model maximizes on the maximum. Okay? In genetic epi data, it's always difficult to describe a likelihood surface, but let's give it a shot. So imagine that you have a likelihood surface with nice clean data with a straightforward underlying genetic model. You can imagine your likelihood surface is a little bumpy, and then you've got one big maximum. Okay? That's the point that you're trying to get to when you're using maximum likelihood methods, is what is the maximum likelihood. With genetic epi data, it seems to be particularly problematic, especially with complex diseases, is that likelihood surface is very bumpy. And so you can imagine your computer's going along, estimating the likelihood. It gets to a hump that's bigger than the rest, and it stops there, and it says, well, here is the maximum. When in fact, it's not. It's a local maxima. And so there are convergence problems that you run into when you're doing these types of analyses, especially for the more complicated models. So we start with commingling analysis, which has relatively few parameters, and you will see that you actually run into this problem even in commingling analysis. When we get to segregation analysis, it gets even worse. But we'll encounter it here first and talk about how to deal with it and what to look for. But this is, yes?
What part of what we've just gone through is actually the commingling analysis?
So the commingling analysis is trying to estimate each of the underlying subdistributions. And so the parameters that I've just described, the means, the standard deviations, and the proportions, each relate to those underlying subdistributions.
And it's called commingling because you have the commingling of the different distributions.
That's exactly right.
Great.
Yeah.
Thank you.
Yes, that's a good question. Very good. So that's right. This is a commingling analysis because we're looking for evidence of these commingled subdistributions. Yeah, the first time I heard commingling analysis, I thought, what is this? But it's what it sounds like, actually. Okay, so to actually test the model, as I said, we fit a series of different models, assuming there's either one distribution, 2 or 3, or on up. And then we look at the likelihood value and use a likelihood ratio test to determine what is the best fitting model. Have many of you used maximum likelihood approaches in other areas? Okay, some. It's something that you don't encounter a lot, and I try to describe a likelihood surface very, well, the biostatisticians I'm sure would have a heart attack, but think of it as a surface. Where you've got the big bump, that is the best estimate given your data and the model that you're trying to fit. And so what you're doing with maximum likelihood approaches is not only getting the maximum likelihood value, but also estimating the various parameters. And the thing that sometimes makes epidemiologists uncomfortable is with a lot of these model fitting approaches, you don't just run it once. You run it numerous times to make sure that you're actually getting the best model possible and the maximum likelihood. So these types of analyses can take quite a long time. You'll see with the commingling analysis is it runs very quickly, but when we get to segregation analysis, which is very similar, it can take days to run. And so it can be quite time-consuming, especially when you need to run about 20 to 50 different models and run them repeatedly. Okay. All right. So what we're doing here, as with most likelihood ratio tests, is we're comparing the log likelihood of one model to the other, and these are nested models, and then we're using a chi-square test to determine whether or not it's statistically significant. You'll see that we try to keep the statistics to a minimum in here. We have enough to do with learning the genetic epi terminology that I really try to keep formulas under control. Okay, the other thing that we need to know when we're doing a likelihood ratio test is the degrees of freedom. Okay, and the degrees of freedom, remember, is just the difference in the number of estimated parameters between our two models. And when we run this, this will make more sense. Now, there is something that I'm just going to mention here, but we're not going to spend a lot of time talking about it. If you do a commingling analysis, there are some references that you should look at, including the NOCOM documentation, which describes this last issue. That these commingling analyses aren't exactly a normal chi-square distribution, and it is most appropriate to correct these statistics using this formula here. However, when you look at commingling analyses that are reported in the literature, nobody ever does this. Okay. Again, remember, keep in mind what it is you're doing with the commingling analysis. I mean, this is not the end-all in terms of analyses, and most people don't bother with this correction. It really does not make a big difference. But you should be aware of this. You may encounter it in the literature, but as I said, most people do not adjust their commingling analyses for this. Okay, so a couple of things about commingling analysis before we actually get there. As I hinted earlier, commingling analysis isn't always the most powerful way to determine whether or not you've got essentially segregation of these genotypes, these underlying genotypes in your sample. The good thing about commingling analysis, though, is very rarely do you get a false positive result. So if there truly are commingled underlying subdistributions, you don't get false positives very often. The issue still, though, is what does that mean? Okay, so you pick up 2 or 3 underlying subdistributions. The issue is still one of interpretation. And remember, it's just that it could be consistent with genetic effects or something else causing variation in the phenotype. Maybe it's measurement error, who knows? Okay, but just because you pick up those underlying subdistributions doesn't mean it's genetic. But there's at least some comfort to knowing that very rarely do you get false positive results using commingling analysis when you compare it against something that in this case would be more of a gold standard, and that would be segregation analysis using family data. Okay. When we get to segregation analysis, this will make a little bit more sense, but at this point you have to trust me a little bit. Okay. And there are some nice papers that I've referenced here if you ever wanted to go look at this. But again, as I said, most people use a commingling analysis for preliminary data, and they don't get too hung up on things like this. Okay. You'd always want to add more evidence to it. Okay, so this is just a little bit more about false positive evidence from commingling. You can look at this article if you'd like. The other issue we talked about is log transformation of the data. Again, think carefully about doing this. It may make it more difficult for you to pick up these underlying commingled subdistributions if you log transform. And in the simulation studies that they did in this paper by Kwan et al., they showed that removing the skewness, in other words, log transforming the trait, had the biggest impact on power of commingling analysis. Okay. So for example, if you used the segregation analysis, which is the gold standard, and had evidence for a single major gene, and then you tried to replicate using the commingling analysis, if you log transform the trait, 20% of the time you were not able to detect evidence of commingling using commingling analysis if you log transform. Okay. And then, now this is something that's a little bit foreign to epidemiologists, but statistical geneticists and some genetic epidemiologists talk about this a lot. Anything that's not genetic to a statistical geneticist is noise. Now, as epidemiologists, we like to think environmental factors are a little bit more important, but frequently in things like linkage analyses and some of the other analyses you're going to do, adjusting for things that are not genetic are done to reduce noise. And essentially what you're trying to do is make the genetic signal as strong as possible and remove variation due to any other influence other than your gene. Okay? So that's something as epidemiologists we have to get used to, that anything non-genetic is noise, and frequently we adjust for that. Okay? There are newer methods now to look for gene-environment interaction, but it's the same issue that we encounter when we're looking for interactions in any situation, is we generally have limited power to pick those up. And it may be that adjusting for these environmental factors will help you to identify your genetic signal. So again, something else to think about when you're thinking about your phenotype. Okay, here's another term for the day. Again, this is all about the phenotype, is if you think about complex diseases and you think about the etiology of a disease and start, let's say at the end where you actually have developed a disease, that's pretty far along a pathway where you have many different things that contribute to the development of that disease, whether it be genetic, environmental, modifier genes, whatever. So if you're really trying to identify a gene that, if you kind of think about the pathway and progression, the gene is way back here, the disease is way out here, there are many, many things that have come in the middle that are going to obscure that relationship. If you can think about phenotypes, quantitative phenotypes, that occur earlier on in the pathway that you know contribute to your disease, you may actually want to think about using these intermediate phenotypes to try and map the genes for your disease of interest. Okay? Sometimes in some areas of genetic epi, these are referred to as endophenotypes. Others, we just refer to them as intermediate phenotypes. Okay? So think about something like cardiovascular disease. We can name a dozen risk factors for cardiovascular disease. It may be that looking at those quantitative intermediate phenotypes or risk factors may be a better approach than looking at cardiovascular disease as your phenotype. Okay. Now, one example that's probably not a good one is blood pressure. People have been trying to map the genes for blood pressure for a very long time, But that in itself is a very complex phenotype. Okay? For diabetes, rather than using diabetes status, yes, no, we may look at glucose levels, we may look at insulin sensitivity, we may look at a variety of other intermediate risk factors. And again, if you're working with a complex trait, that's something else you should be thinking about. And commingling analysis is very good, or is very useful for trying to screen a number of these intermediate traits. And the reading that's listed for this lecture actually describes such an approach, where they're looking for using commingling analysis to try and screen out several intermediate phenotypes that people have suggested are genetically influenced. And you'll see in the paper that that's may or may not be the case. Okay. All right. That's the paper I'm referring to right there from the Rochester Family Heart Study by Sharon Cardia. It's a very nice paper on commingling analysis. It gets at many of the issues of thinking about your phenotype and adjusting for different covariates and considering a panel of potential phenotypes to look at. Okay, so to summarize before we jump into the demonstration, commingling analysis can have low power, which limits its usefulness. One of the biggest things that influences the power of the commingling analysis is the skewness in the distribution. So log transformation has a big impact there. The good thing is that when you have positive evidence for commingling, we very rarely get false positive evidence for underlying subdistributions if they're not there. The usefulness of commingling analysis is it makes you think about your phenotype. It also can provide you guidance in terms of selecting from several possible phenotypes to use. And it can also be used as preliminary evidence for genetic influences on a particular trait. Okay, so let's go to the demonstration. Let's keep our fingers crossed that this works. Okay, so the first thing you'd have to do is go to the Rockefeller website and download the software program for commingling analysis. This is actually found in, they have a series of different pieces of software, and the commingling analysis is included in the one that says linkage utilities. So in the linkage utilities package, there are a variety of different softwares, including commingling analysis, a little module for doing Hardy-Weinberg equilibrium, and a variety of other kind of basic things that you're going to do with genetic epidemiologic data. Okay, so you'll get several different packages. What you will get with the commingling analysis is the executable file. You will also get a data file called nocom.dat that you can use to run to practice doing the commingling analysis. And that's what I'm going to run today. The nice thing about doing that is you can look at the documentation and you can look at the output file that they give you to make sure that you're running it correctly and you're getting the right solutions. So it's actually very nice. Okay, so download the package, you'll get the data set. It's a simulated data set. You'll get the output, and then, as I said, you can practice running commingling analysis. Every year I do this, it takes me a couple of times to run it because it's not intuitive right away. Like most of the things. So keep in mind that what we're trying to do with commingling analysis is fit a series of normal distributions. The only parameters that we have to worry about are the means of each of the underlying distributions, the standard deviation, and we're going to assume the same standard deviation for each subdistribution, and then the proportion of individuals in each of those subdistributions. Before you do a commingling analysis, what you should do is plot out your data. Look at the distribution of the data. Okay? Use a histogram, do something, plot it out, and in your mind, visualize what we did up here is if you were going to fit a series of distributions to this data, where would they fall? And then use the very accurate eyeball test to estimate what the mean of each of those subdistributions would be and what the standard deviation might be and approximately what proportion of your subjects fall into each of those distributions. With all of these maximum likelihood approaches, it's best if you give the program starting values. Okay? You know that those are not going to be the final values, but give it something reasonable to start its estimation with. Okay? And then, as I said, you're going to rerun the program multiple times to make sure that you get the same solution. And what you do is it's an iterative process. You give it the starting values, you let it run, it gives you better values, you plug those back in, you run it again to make sure that you arrive at the same final solution. Okay? But you do have to give it something reasonable to start with. Now, I've already looked at the histogram, and I've used this example for a number of years, so I know approximately what some good starting values would be here. But you guys should look at the data first before running the commingling analysis. Okay, so you call up the NOCOM executable, and the first thing it asks is whether or not you want to write this to a file or not. So I'm not going to write it to a file because I already have a file. So I'm going to say no, don't write it. So the first thing it asks you is whether or not you want to transform your data. We're going to say no. We do not want to log transform our data. Okay? So you put in a value there to use the data as it is. You just give it a 1, which is no transformation. And it asks us if we want to estimate this exponent, which we don't since we're not putting one in. And then it asks you how many components do you think underlie your data. So when it says components, it means how many subdistributions do you want to model. With most of these maximum likelihood approaches, you start out with the simplest model first, and then you build up. Okay? This is especially important when we get to segregation analyses, where you're estimating lots and lots of parameters. You start with the simplest model, get good estimates of the limited parameters in that model, and then build up. Okay? So with the commingling analysis, the simplest model is going to be a single distribution. We're saying, just model it as one distribution. Tell me what the mean is, tell me what the standard deviation is, etc. So that's what we're going to do right now. Now, that ran, it's done. As I said, this is a really fast little program. Okay? So this, I don't know what you can see there, but essentially what it gives us is, this is the mean for the first distribution, and it says our mean is 11.16. Standard deviation is 3.07, and obviously the proportion of subjects in that distribution is 100%. So we're saying it's a single distribution, everybody's in that distribution. The other key piece of information is your log likelihood, which is what you're going to need to compare your models. So in this case, the log likelihood is minus 324.4, blah, blah, blah. Keep in mind with these likelihoods, when the value is negative, a better likelihood is closer to 0. Okay? It gets a little confusing here, but as with many of our models, the more parameters you get in, the better the likelihood should be, even though it may only get better slightly, not necessarily significantly, but the more parameters you estimate, the better the likelihood. So one of the important things that you need to look at when you're doing commingling analysis is, as you add more models, more parameters to the model, does the likelihood get better? It doesn't have to be significantly better, but you should see that likelihood value changing and getting closer to 0. Okay? If it's not, that means you're probably on a local maxima and you need to rerun it again. And that's one of the tricks that you have to look for with this program and many of the other maximum likelihood approaches. Okay, so now we've got our values for a single distribution, and if I were doing this, I'd have myself a little piece of paper with a table that says, the means, mu1, mu2, mu3, common standard deviation, the proportion F1, F2, F3, and then the likelihood of each model. And then I'd write these down, or you can write it to a file. But I kind of like to write it down just so I can see it as I'm running it and compare without having to go back to another file. Okay, so let's run this again now. We're going to estimate 2 parameters. So again, it asks us for the exponent, and we say no, we don't want to transform the data. We don't want to estimate that. And now we're going to say we want to estimate 2 subdistributions. So it says enter number of components. We just enter 2 here. Remember, we go from the simplest to more complex. Now, this is where it gets tricky, and I have to read and pay attention here because now it asks us for the standard deviations, and it says, do you want to estimate separate standard deviations for each subdistribution, or do you want them to be the same? We want them to be the same, and it's a little tricky here because the only way you can make them be the same is to say it asks for the ratio of the standard deviations. So we're going to say the ratio is 1. Okay. Now it's asking us, now that it's determined it's only going to estimate 1 standard deviation, it's going to go through and ask us about starting values for each of the parameters for each of the subdistributions. So it starts with the first subdistribution, and we're going to be asked about what's our idea of what the mean should be, standard deviation, and then what proportion of subjects is going to be in that first subdistribution. So let's, again, I should have shown the histogram here, but I didn't want to risk doing too many fancy things. So we're going to put in as the starting value for our first subdistribution as 10. And we want to estimate the mean. So remember, we're just giving it a starting value, and we want it to estimate it to a more accurate value. And then the standard deviation I'm going to put in as 4. And we want to estimate the standard deviation. This is where it's a little confusing. It asks for the weight or the probability of component 1. What that means is what proportion of subjects are in component 1. I don't know why it doesn't say proportion, but it asks you for a weight, which kind of throws you off. The easiest thing to do if you haven't eyeballed it is just to put in, when you're doing 2 distributions, is equal percentages in each. So we're going to say 0.5. The thing about commingling analysis is if these don't add up to 1, the program will crash. So you kind of have to keep track of it, especially as you're doing more subdistributions. It throws you out and you have to go back and start all over again. So this is where you have to pay attention. Okay, and then we're going to estimate that proportion because we don't think it's really 0.5. Now it's put in all the parameters for the first subdistribution, and now we're going to go to the second subdistribution. So it asks us for the mean. I'm going to put in 15. We want to estimate that standard deviation for component 2. Estimate the standard deviation, okay. And then we have to, it's asking for the weight of component 2, so we have to put in 0.5 because that's what we put in for the first one. Estimate that, oh, good, it ran. Whew. Okay. So you can see this is relatively clean data. It's simulated data, so it's not real messy. And this ran pretty quickly. And so what you look for is you go down to the last line, that's your final solution. And this just goes through and shows you the iterations it went through to get to this. And what you can see is this column right here relates to the first subdistribution, and this column relates to the second subdistribution. So you can see here that the mean for the first subdistribution was 10.5, standard deviation of 2.4, and it says 90% of the subjects are in that first subdistribution. Now the second subdistribution, the mean is 17, and I did something wrong because it didn't give us this common standard deviation. I put in the wrong number at the beginning, and it says 9% of the subjects are in that second subdistribution. And then the likelihood, maximum likelihood value was minus 313.41. What did I say the likelihood was for the other one? Minus, what was it? 34. That sounds about right. Okay. So you can see that this likelihood is closer to 0. This is a better likelihood. The way we would determine whether or not this model fit better than the single model is to compare the 2 likelihoods. Okay. Multiply them times 2. So we're going to do this roughly in our heads. If it was 3, let's say it was 333 approximately. So the difference between those 2 likelihoods is 20 times 2 is 40. Okay. This is a chi-square with 2 degrees of freedom. That's going to be statistically significant. So we say that the 2-mode model fits the data better than the single mode. That's good for a genetic epidemiologist because now we're saying that there are at least 2 subdistributions that fit our data better than a single subdistribution. So that could be consistent with either dominant or recessive effects, but at least it's not polygenic. Okay? So then we'd go on and we would do the same thing. Let's try to fit a 3-mode model and see if I can do it right this time. So you can just start over with the same data. We don't have to go back out of the program. So again, we're not going to log transform it. Number of components, let's try 3. Fixed ratio, and what did I do last time? Let's see if I can do it right. That should be 2 this time. Okay, mean for component 1. I'm going to put in 10 again. Estimate this mean. Standard deviation, let's say 3. This is where it gets tricky because now we're going to have 3 subdistributions and we have to keep track of the proportions so they add up to 1. So let's do 0.2 for the first one. We're going to estimate that. Now it's asking for the mean of component 2. So let's do 13. We're going to estimate that. Ratio of standard deviations, there we go. So we're just going to say 1. So the ratio of the standard deviation is 1, so that says the same standard deviation. Now it's asking for the proportion of individuals in component 2, and let's do that as 0.4. So we've got 0.4 and 0.2, so that's 0.6 so far. Mean for component 3, let's say 16, estimate that. Ratio of standard deviations is 1. Weight of component 2, so that's 0.4, 0.2 is 0.6, so 0.4. And then we're going to estimate that. Estimate standard deviations, there we go. Okay, so here are the 3 underlying subdistributions, 1, 2, and 3. Their mean values, what is that? The first mean is 8.8, standard deviation of 1.8, and 37% of the subjects are in the first distribution. Second distribution mean is 11, same standard deviation. 43% of the subjects are in that distribution, and then the final distribution mean is 15.9, same standard deviation, and 18% of subjects are in that distribution. And the likelihood for this model is minus 314. So the likelihood, what was the likelihood for our other model? 314. So this is not significantly better. It's slightly better, but it's not significantly better. So we would conclude then that the 2 distributions fit the data better than 1, 2 distributions fit the data better than 2, and we would say that we have evidence for 2 underlying subdistributions in our phenotype, which, as I said, could be consistent with either dominant or recessive mode of inheritance if it's a single major gene. Okay, so you guys now have one tool. You can run commingling analysis. It's, as I said, not a bad one to run. If you're looking at the website tonight and you go to Rockefeller, you might want to download the NoCom package and just try running it while it's fresh in your mind. Their documentation, as I said, is actually very good compared to many of the other packages out there. So you should be able to run the commingling analysis on your own. Okay, well that concludes our first day in genetic epidemiology, and that concludes our lecture on commingling analysis, running the software package, and starting to think about your phenotypes. Okay, thank you.